Okay. So students, I think last class was on antigen and antibody. And you know what an antigen is? It is any foreign substance entering the body. And antibody, it is the protective mechanism produced in the body against any antigen. It could be either a microorganism or any foreign particle which can enter the human system. And so today we'll be looking into antigen and antibody reactions and also about the complement, which is a non-specific immunity which occurs in every individual. So coming to antigen and antibodies, they combine with each other specifically and in an observable manner. Specific and observable manner are the main key points in this antigen antibody reaction. And these antigen antibody reactions, they can serve several purposes. It is used for several purposes, out of which they form the basis of antibody mediated immunity and also hypersensitivity and any autoimmune disease. Apart from this, this antigen antibody reaction is highly useful in the laboratory in the diagnosis of various infections and also in epidemiological survey. Now, as in COVID-19, they look for the antibodies as an epidemiological survey, which is usually a serological test, which is mainly based upon the antigen antibody reaction, which is occurring in the laboratory setup. And coming to antigen antibody reaction, an antigen and antibody reaction occurs only in the presence of electrolytes at a suitable temperature and pH. So you need these three factors: electrolytes, suitable temperature, and pH for an antigen antibody reaction to take place. And there are two types of antigen antibody. So where did I start? From where did I? From where I was? This slide. Hello. Hello.
Hello, ma'am, from starting only.
From starting.
Yes,
Okay.
ma'am.
Okay. Hmm. So now I am audible.
Yes, ma'am, we can hear
Yes,
you.
ma'am.
So antigen antibody we have already dealt with, and now we are going to antigen antibody reaction and the complement. So any antigen antibody can combine with each other specifically and in an observable manner. So the two key points in this antigen antibody reaction is the specificity and in an observable manner. Why this observable manner is because most of this antigen antibody reaction are used in the laboratory setup, and we need to report based upon the positive or agglutination or precipitation. And this antigen antibody reaction, they serve several purposes, like they form the basis of antibody mediated immunity, hypersensitivity, the pathogenesis of autoimmune disease, etc. And they are very much useful in the diagnosis of various infectious pathogens and in epidemiological survey. Epidemiological survey, you know very well. Now ICMR is doing an epidemiological survey for COVID-19, where they are detecting specific antibodies against COVID-19. So this is also an antigen antibody reaction which is very useful in the epidemiological survey. And the antigen antibody reaction can occur in the presence of three conditions. One, the presence of electrolytes, and then a suitable temperature and pH. And there are two types of antigen antibody reactions, namely precipitation and agglutination. The basic difference in precipitation and agglutination is the nature of the antigen. You need a soluble antigen for precipitation to occur and a particulate antigen for agglutination to occur. So you can convert precipitation to an agglutination by coating the soluble antigen onto a carrier particle. And precipitation is very, very sensitive for detecting antigens, whereas agglutination is very, very sensitive for detecting antibodies. Okay. Am I audible?
Yes, ma'am.
Okay. And when does this antigen antibody reaction occurs in an observable manner? There are three phases of this antigen antibody reaction. The initial phase is the prozone phenomenon, where there will be an antibody excess, and then the later half you will have an antigen excess, and the middle part, which is the zone of equivalence, where there will be equal number of antigens and antibodies only. You will see, or you can see the antigen antibody reaction in an observable manner. And usually the antigen is usually multivalent, or the epitopes are numerous on the surface of the antigens, but antibodies are only bivalent. It has only two antigen binding sites. And this antigen antibody ratio is very much important for the agglutination or precipitation to occur in an observable manner. So this here you can see that there are equal number of antigens and equal number of antibodies. So it has formed a film or lattice so that it can be visualized in an observable manner. And coming to precipitation reactions, as I once again I want to repeat, precipitation reactions are highly sensitive for detecting antigens. There are various types and methods of precipitation reaction. One such is a ring test, where you add a solution of the antibody and a solution of antigen, and they mix together, and at the junction, the zone of equivalence, they form a precipitate, and that is called as the ring test. And the classical example of ring test is the Ascalis thermoprecipitation and Lancefield classification of streptococci. How many types of Lancefield classification of streptococci exists? Anyone? Anyone?
15.
21. Without ink. Okay, so they are based upon this is mainly based upon the surface component of the streptococci, where we do this Lancefield classification. Then we have slide test, which is the VDRL. What is the abbreviation of VDRL? Abbreviation? Venereal.
Ventral disease research laboratory or
It
something.
is done for syphilis. Okay, this is done in a slide, so it is called as a slide test. And tube test, you have Kahn's test, which is also a test done for syphilis and for standardization of toxins. These toxins are mainly used in the vaccine field of vaccine. Then you have immunodiffusion. Immunodiffusion means there are two phases, one a phase in the air and other in a gel. So this gel, it is a semi-solid agar-like substance, where it has spaces in between, which can allow the antigen or the antibody to freely pass through. This property of the gel is used for this immunodiffusion test. There are various examples like radial immunodiffusion, counter immunoelectrophoresis, and rocket electrophoresis. And this is rocket electrophoresis, where this is the gel. The gel will contain the specific antibody. Say you take this COVID itself, you have specific antibodies incorporated into this gel, and these are wells which are created with templates. And when you add the antigen into it, the antigen diffuses into the gel, and at the point of equivalence they form a precipitate line. This has a dye, so this dye will make it visible on the site of precipitation. And this is the standard. The standards are the one based upon which we compare the amount of antigen in the specimen which you have added. So this is based upon the height from the center of the template to the peak of the template, and usually these kits will have the standard values, and based upon this height you can identify the amount of antigen. You can do quantification of antigens in this rocket electrophoresis. This is the radial immunodeficient, where you have the template here. The same way the antibodies in the gel surrounding, the antigen will diffuse and it will form a zone of precipitation. And based upon the diameter, based upon the references given, you can identify or quantitate the amount of antigen in the sample. That is immunodiffusion. That is regarding precipitation reactions. And coming to agglutination reactions, agglutination reactions are more sensitive for detection of antibodies, and you have two different types, namely the slide agglutination and tube agglutination. Slide agglutination, the classical example is blood grouping and cross matching, and tube agglutination, classical examples are Vidal and serology for brucella. And in agglutination, you have another type called as the heterophilic agglutination test, where we use, usually what we do, as I said in the first point, specificity for antigen antibody reaction is a must. An antibody which is specific to the antigen can only react with the specific antibody. Is it? There are certain conditions where instead of using the antibody specific to the antigen, we use another antigen because of cross reaction. Say for example in Weil-Felix test, which is done for typhus fever. Typhus fever is caused by a rickettsial group of organisms where cultivation of rickettsia is very much difficult and it is not cultivable so easily. So availability of the antigen is very less. So they have a cross-reacting antigen with Proteus vulgaris. So that property is utilized. So the antigen of Proteus vulgaris is used to detect the antibodies produced against typhus group of rickettsial diseases. The same way Streptococcus mg and cold agglutination test for primary atypical pneumonia, where the heterophilic agglutination is mainly based upon the cross-reacting antigens. And Coombs test, there are certain antibodies which are called as incomplete antibodies because though they attach themselves to the antigen, they will not produce a visible precipitate or agglutinate. So in order to make them visible, we introduce an antibody to the already attached antibody and you look for the reaction based upon an indicator system. That is called as the Coombs test. And passive agglutination is nothing but conversion of a precipitation reaction to an agglutination reaction with the help of latex beads or else bentonite or RBCs. You can use many number of things to convert precipitation reaction to agglutination reactions. And there are in the Coombs test, you have two different types, direct Coombs test and indirect Coombs test. So Coombs test is mainly done for detecting incomplete antibodies. This incomplete antibodies is a classical picture of RH incompatibility. So the basic example for direct Coombs test is the baby sample which is positive in the presence of mother's antibody on the surface of RBCs in erythroblastosis fetalis. So this sensitization, that is the antibody attaching to the RBC, occurs in vivo within the human system. So you have an antibody to this antibody, that is anti-human immunoglobulin, which is called as the Coombs reagent. So this will now agglutinate or attach to the antibody, incomplete antibody, and it will produce agglutination. This is called as direct Coombs test. Whereas indirect Coombs test usually happens in vitro. That is in the laboratory. We do a prenatal testing, which is usually done to determine the presence of non-agglutinating antibodies in her bloodstream, which can attack the fetal RBC when the father is RH negative, RH positive, and mother is RH negative. So you take the RBC of the mother and you and the patient sample, and then you mix it this. So this happens in vitro in the laboratory, and then you add the anti-human immunoglobulin which produces agglutination. So the basic difference is the sensitization of the incomplete antibody with the antigen. If it happens in vivo, it is direct. If it happens in vitro, it is indirect Coombs test. There are others like complement fixation test, which is called as the Wassermann's test, which I'll be explaining now. Neutralization test for viruses and toxins, and immunofluorescence. There are certain fluorescent dyes which will take up light in one wavelength and emit light in another wavelength. So this property is used for tagging the antibody with the fluorescent dye to detect various antigens. The dyes which are commonly used are auramine, rhodamine, and fluorescent isothianate. The same way, radioimmunoassays are also available, where instead of the fluorescent dye, radioactive isotopes like iodine 131, etc., are tagged to the specific antibody, and then they are visualized under a the radioluminance meter, where you can look for the radiance of the radioactive isotope. Coming to complement fixation test, this complement, as I said, is a non-specific component present as an innate part of immunity in every individual. One of the main functions of this complement is lysis of cells. That property is utilized in this complement fixation test. Say you take a serum which contains the specific antibody and the antigens are added. So this antigen and antibody will combine. Then the complement is added. Complement is usually stimulated only by an antigen antibody complex in case of the classical complement pathway. So once this complement attaches to the antigen antibody complex, then you add a indicator system. This indicator system is nothing but sheep red cells which are already sensitized to this antibody. Sorry, this antigen. So this is used as an indicator. When you add this, what happens is this complement is already utilized in the antigen antibody reaction which has happened earlier. So the RBCs will not lyse. So because the main final end product of complement activation is cell lysis. But this will not happen because the complement has been already fixed by the antigen antibody complex in case of in the previous step. So this indicates that the reaction is reactive and the patient sample contains specific antibodies. Say the patient sample does not contain antibodies and the antigen which you added, so the antigen is free, so the complement which is added will not be activated. Now when you add the indicator system, the RBCs will have the free complement and the complement will now stimulate lysis of the RBCs because it is a sensitized RBC. Sensitized RBC means the RBC which has been already coated with the antibodies. So this is an antigen antibody. So now the complement can attach and it will cause lysis. So lysis means a negative reaction. No lysis means a positive reaction indicating the presence of antibodies in the sample. Okay, do you follow? Hello?
Yes, ma'am.
Okay. Any doubts regarding this?
No, ma'am.
Okay. Then you have ELISA. ELISA, you must have left and right known about it. The same principle. This is a hard well, and you have the antigens coated in the well. If you want to detect the antibodies, if you want to detect the antigen, the antibodies can be
Some
coated.
students are watching through the YouTube, madam. Actually.
Okay.
Okay.
Okay. So this antibody, when you add the sample which contains the antibody, this antibody will bind to the antigens which are fixed on the well. And then usually this ELISA, it has multiple washing procedures to remove the excess of antibody. So these two excess antibodies have been removed. And next step is adding an anti-human antibody which is tagged with an enzyme. This is also washed to remove the excess. And finally, you add a substrate. This substrate is where this enzyme will act upon and produce a color change. So based upon the color change, which is usually measured in terms of optical density, you get a cutoff value and you give a quantitation of the antigen or antibody present. This is the ELISA. Now, ELISA is available for most of the infectious agents, cancer agents. Many applications have been using this ELISA principle for detection. Okay. And next you have the blotting techniques. Blotting techniques are nothing but where you have a filter paper or a gel where you separate the components of 1 second, sir. Okay, sorry, sir.
Okay, madam, go ahead.
Okay. So a blotting technique where you can blot the filter paper or in gel, either the DNA it is called, if it is the DNA it is called southern blot, RNA it is called as northern blot, and proteins it is called as the western blot technique. So this is the western blot which is mainly done for HIV where the different proteins have been separated. And coming to the questions, which of the following is included in the indicator system of a complement fixation test? Specific antibody and complement, specific antigen and complement, red blood cells and hemolysin, guinea pig serum. Which one? A, B, C or D? Anyone? It is C, red blood cells and hemolysin, which is the indicator system where the RBC, which is with the specific antibody, has been coated. So the complement will attach to this antigen antibody complex and cause lysis of the cell. So lysis indicates a negative test and no lysis or hemolysis indicates the positive test. Which is not a heterophilic agglutination test? Bifilix, Vidal, Paulbanal, Streptococcus mg. Heterophilic agglutination means detect mainly using the principle of which one? A, C or D?
C.
C. It is B, Vidal test, which is a tube agglutination test. So heterophilic means common antigen sharing. Prozoon phenomenon is a feature of tularemia, Legionnaire's disease, plague, brucellosis. Which one? It is brucellosis because brucellosis is the classical example of prozoon phenomenon. When you suspect brucellosis, the sample of the patient has to be diluted many folds and only do the test. Only then the zone of equivalence will be there for detecting an observable antigen antibody reaction. So that is why you undergo serial dilutions have to be done to identify the titer of antibody. The following methods of diagnosis utilize labeled antibodies except ELISA, heme agglutination inhibition test, radioimmunoassay, immunofluorescence. ELISA,